It's a little ridiculous. Okay, so we're going over carbohydrates. Yesterday when we talked about lipids, right, we started off by basically crossing off the monomer, the structure, the function. There was no common structure, function, monomer. Now with carbohydrates, and again when we go into proteins and nucleic acids, we are going to have that kind of structure, and I mean that more in learning kind of way. So carbohydrates do have a monomer. The monomer itself is a carbohydrate. The polymer is a carbohydrate. What is the monomer for a carb? Monosaccharide. Mono meaning one, and sac, that S-A-C-C-H, meaning sugar or sweet, right? So a monosaccharide. And there's a bunch of different monosaccharides. We're going to break them down and look at it. The structure is just a repeating unit of monosaccharides over and over again. And mainly the function is going to be used for what's the function? Why do we eat carbs? Yeah, Prince. Yes, it's going to be your main source of energy. Whatever you don't use, you'll store, which means if you eat a lot of sugar and fat at the same time, you're going to burn the sugar first. The fat's going to be saved. You don't convert sugar to fat. You don't convert sugar to fat. They're different. There are chemical processes that will do that, but for the most part, you'll burn the sugar, save the fat for later. Okay? We can be looking for carbohydrates at simple sugars. That's going to be your glucose, etc., or polysaccharides. Note, anytime you see OSE, you're thinking sugar. You're thinking a carbohydrate. And we can refer to monomers and to the dimers, the smaller pieces, as simple sugars, the bigger ones as polysaccharides, and potatoes are not simple sugars, they're polysaccharides, so it's funny. Haha, haha, no. Moving on. All right. So when we take a look here at the classification of different monosaccharides, two things are going to be important. Come back up here. First of all, where's the carbonyl? Remember, the carbonyl is going to be the C double bonded to O. And so if that C double bond to O is at the terminal end, this is going to be an aldehyde. If it's in the middle, that's going to be a ketone. You might see those words. I would never expect you to necessarily memorize that, but I simply want you to understand that that carbonyl group can shift within the molecule and make completely different isomers. And then we're also going to name sugars and classify them based on how many carbons they have. Now, sugars are going to follow the same general formula, right? Carbon, hydrogen, oxygen in a 1 to 2 to 1 ratio. However, the number of carbons can change. Up here, if we have 3, what is something like 3 usually prefixed with? Tri. So this is going to be a tri, and it's a sugar, so we're going to call it a triose. Okay, how about 5? Think of a place in DC. Penta. So this becomes a pentose. And 6? Hexose. Okay. They're all following the same 1 carbon, 2 hydrogen, 1 oxygen formula. They all have a carbonyl group somewhere in them. But the number of carbons can vary. The location of the carbonyl can vary. So far, so good? All right. So an important thing that we have to know is that in crystalline form, you can end up with this kind of linear structure. But most of the time, when you put sugar in water, a good chunk of it is going to form into what is called a ring. a ring for obvious reasons because it loops back on itself. But we've got to know how to number. This sounds stupid. It sounds really asinine, but you do need to know how to number these because we're going to come back and see when it comes to how they link, it's named based on the numbers. When it comes to how different DNA strands come together, there's sugar in DNA. So you have to know how it's numbered. So let's go ahead, and we're always going to start at the carbonyl. So here's our carbonyl. It's the terminal one here. And we're going to number straight down. So this guy here, any guesses as to what the first carbon will be called? Carbon 1. Okay. Okay. That was luck. Let's see if we can figure out the second one. Carbon 3, right, Printon? Carbon 2, then carbon 3, then carbon 4, 5, and 6. You'll see that carbon 5 has an OH group that's diagrammed with blue. We're going to want to keep an eye on that because when the ring structure forms in hexoses, in 6-carbon sugars, usually it's going to be this OH right here that is involved in the bond. So this OH here kind of wraps back around to the top and makes the bond. So again, let's go ahead and number. I need to find my carbonyl right here. And then go ahead and number them. 1, 2, 3, 4, 5. And then do I label my O as 6? No, I'm numbering carbon, so I have to go outside. We've got one kind of floating off to the side. So carbon 1 is at the carbonyl, the C double O. The carbonyl group. Okay. Okay. Once the bond has formed, again, you're left with 1, 2, 3, 4, 5, 6. Somebody asked earlier if it's always clockwise. No, I could just as easily have something that looks like this. And it's not going to be clockwise. I'm going to start at the carbonyl, which is this guy, and go around. An easier way to remember it is start at the oxygen. Think of oxygen as 0, and then just go around whichever way will allow you to connect all of the carbons. So if I start in blue, like if I was to start, is this 0, 1, 2, 3, 4, 5? I exclude this guy, so that's wrong. But that's right. Ashley.
Does that explain why the blue it's blue from the fifth?
Yes.
Is that what branches off?
Exactly. So it's just trying to show you that it's that O that's there right here from the 5 carbon that becomes this O that becomes part of the ring. All right. So who cares? Well, you should, because here we go. In solution, it's not just going to form one type of ring. There's actually going to be 2 rings that form from glucose. So I've got a glucose linear in the middle here. But glucose can form 2 types of ring. That carbonyl group can end up facing downward or facing upward. And that makes a huge difference. If the carbonyl group, the O there, right here, is facing downward, we're going to call that an alpha glucose. If it is facing upward, that's a beta glucose. And remember, this would be a 3-dimensional structure. So on the plane, like if you were to look at the ring and turn it kind of up like this, then it's down versus up. But you'll never actually see it in 3 dimensions. Okay. How are you going to remember that? Because you do need to remember alpha versus beta glucose. You do need to remember how that forms, right? This carbon hanging out there. How are you going to remember alpha versus beta? Well, what's the famous yoga pose that everybody knows?
Downward dog.
Downward dog. So I think of downward dog as the alpha dog. And so the alpha is down. And for beta, it's just the other one.
I don't think that helps though.
So alpha is down.
Oh, 'cause alpha dog.
Alpha dog.
Oh my gosh, that's
Downward
so good.
facing dog, alpha dog,
Wow.
right?
That's
So
a good
like,
one.
because I'm weird. So now it's just a completely sugary dog. Whatever. I'm having too much fun with my iPad. All right, so why do we care? Again, this doesn't matter, does it? Whatever. I took a linear thing, I made it a ring, and now there's two kinds of rings. And what's your dang point? Well, this is where it's important. Yeah, look.
It makes disaccharides different, right? Because how the way
It does.
carbons connect
And
to
it's
each
not
other.
just the disaccharides that are going to be different, it's going to be the polysaccharides.
So, like super.
Side conversations, hold up. Okay, I want to start with the alpha. Okay, I take my alpha carbon, my alpha glucose. I take the one carbon, and that's going to link to the 4 carbon OH of an adjacent molecule. And so what I end up with is 1, 2, 3, 4, 1, 2, 3, 4, is a bond called a 1,4 alpha linkage. An alpha 1,4, and it's called a glycosidic linkage. Yesterday we looked at lipids. It makes an ester bond. This time it's making a glycosidic bond. It's just a fancy way of saying, look, it's a covalent bond. Ooh, I gave it a fancy name because I'm a scientist and I gotta make things complicated. You're welcome. Ashley, go ahead.
Does it still like produce an H2O or something?
Yes.
Okay, because that's it. That hurts and stuff.
Yes, you're still going to pull out that H2O. It's still a dehydration reaction or a condensation reaction. So this is really just showing you that when you've gotten a whole bunch after that reaction is done, you end up with this structure. And it's kind of got a helical nature to it. So it kind of spins around in little helices.
Is the reason cellulose so strong because it doesn't have that bond like they do? It's like a 1.
It's a
2
different
by
type
2?
of bond. It's still a 1,4.
But it's
It's also it's a different type. Let me pop on that one. Kind of.
They worded it in the book like that, but they said it was something else.
Yeah. We'll look at that one in a sec. So when we're looking at the alpha, all organisms have the enzyme to convert simple sugars to make alpha 1,4 glycosidic linkages. So if you are a plant and you've made a ton of sugar, and now you need to store some, right? It's time for fall and winter, and you need to make sure that you have enough extra energy stored. You're going to store that as starch. There's 2 types of starch, amylose and amylopectin. I don't need you to know that. Simply that plants are going to store their extra energy as starch, which are made out of alpha 1,4 glycosidic linkages. And you and me, let's say I eat a ton of starch. I love potatoes, so I eat a whole bunch of that, and I break it down so that each of these individual sugars here is now in my blood, right? I've raised my blood sugar. My body says, let's lower the blood sugar, let's save the stuff that we don't need for now and use it later. So instead of saving it as starch, we're going to save it as what molecule? Glycogen. And glycogen, in addition to having those alpha 1,4s, is also going to have alpha 1,6. Okay, so that's going to cause these branch points that we see. So you can see the helical nature right here of just the alpha 1,4s, and then it branches off right here. That's going to be due to an alpha 1,6, where a 1 forms with the carbon 6 and kind of zigzags off to the side. Rowan, did you have a question?
Yeah, I still don't understand why, like in this one, there's like the hydrogens connected to the OHs, but in the other, like the bottom one, it's not.
Oh, good question. In this picture, why are there no hydrogens? Because it takes up too much space to show them. They're still there. It's just
not red.
they're just ignoring it, pretending it doesn't exist for now because it's not involved in any of the chemical reactions.
Okay.
So it's just like, whatever, it's there.
Okay.
Good question. Okay. All right. So who cares? We're still there. Who cares? You can convert from polysaccharides to monosaccharides and back to polysaccharides. Great. Well, if you're a plant, you also have a really cool enzyme that can use beta glycosidic linkages. So here's our beta glucose. And here with the beta glycosidic linkage, notice that it's going to be taking an upward facing OH at the 1 carbon to a downward facing OH at the 4 carbon. So what you end up with is more of a sheet nature where one glucose is, that 6 carbon is facing one direction, and then in the next glucose, the 6 carbon is facing the opposite. So it flip-flops, up, down, up, down, up, down, up, down, etc. And that's part of what's going to make it strong. It's also going to make it more linear. It doesn't spiral around. So now there can be additional interactions between 2 adjacent strands of this beta glycosidically linked glucose. Ashley.
Why does that happen for beta glucose and not alpha?
Because alpha just kind of makes it spiral. Almost think of it like,
But why?
trying to think of like a good analogy. With alpha, think of it like this. If I have this shape and I just continue to draw more and more of them, they have to kind of spiral, right? If I'm doing it in a beta, but I'm taking that same shape and going in opposite directions, I've got one that goes this way, one that goes that way, one that goes this way, one that goes that way. So it never really spirals. Instead, it kind of makes more of a flattened out shape.
But like, is it for a reason, like different functions for them? Like,
It does end up functioning differently.
Okay.
Yes.
I just didn't know what made
Yeah, so the
beta
reason
better.
beta's not so
Straight
much
line.
better, but that straight line allows for these adjacent cellulose fibers to also bond next to each other. So this is going to increase the strength, the tensile strength of cellulose. But you also don't have the right kind of enzyme to break down beta linkages. Beta linkages cannot be broken down by mammals. But you say to me, but then how does my bunny get fat when it eats celery? How do bunnies break down cellulose? How do bunnies break down cellulose? Trick question. They don't. They have bacteria in their guts that do it for them. So they have a mutualistic relationship with the bacteria that live in their guts. Also, termites have that. So if you were to sterilize a termite and like get rid of all of the microbes, it could no longer digest wood.
Would it get fat then?
Nope. It would die because it wouldn't get any nutrients. The only thing that it can eat is wood, so it needs the microbes to be able to give it the nutrients that it needs. Okay? So beta glucose gets used to make cellulose, alpha glucose gets used to make starch if you're a plant, glycogen if you're an animal. A lot of information kind of thrown at you at once. How are you doing so far? Okay, we talked plants, we talked animals. We have one major multicellular classification left that we've not discussed. They're my favorite. What are they? Fungi. Fungal cell walls do not use cellulose. Instead, they use chitin, and it's not called chitin, it's called chitin, like a kite. Okay, chitin. So chitin has just this little extra nitrogen containing moiety there. And not only do fungal cells use it,
That's a kite. So kite.
in addition to fungi using it, so do animals. Animals use it as their exoskeleton. In other words, when you squish a bug, what you're really breaking is the chitin bonds and squishing its intestines. Okay? What does that tell you about fungi and animals? We've got a common ancestor, right? And that branches off to make two lineages, one that can make chitin, one that cannot. The one that doesn't is plants. And that's why we know that animals and fungi are more closely related to each other than either one is to plants. Phylogeny. We're bringing it back. Kicking it old school or something. Guys, I'm hilarious. I hope you watched this video tonight and you're like, oh, she is so funny. 10 o'clock me is not... Yeah, it's recording. Say hi.
So we have to
I hope so. Because otherwise... Yeah.
Good question.
Girl, I got you. I hope.
Oh wait, your voice isn't gonna be.
If you hear me, then Maggie was wrong. Okay. All right. With that, let's go over a quick overview. Monosaccharides are single sugars. Disaccharides are 2 sugars combined. Polysaccharides are a whole crap ton of sugars. Polysaccharides that we're worried about are all composed of glucose. That says alpha or, that's like the ugliest. Yeah, there we go. All right. I would expect you to be able to name an example of each. And if I gave you an example of a monosaccharide, I would expect you to know that it's a monosaccharide. So here's the ones that I want you to know. I want you to know the hexoses. I want you to know the disaccharides. And I want you to know glycogen, cellulose, and starch. Okay? That's 9 different things you need to know. I'm going to give you some mnemonic devices to remember them. Okay, your monosaccharides, glucose, galactose, and fructose. They are isomers of one another. Stereo and structural isomers of one another. Glucose is going to be the most important biologically for us. That's what we need to be able to do what's called cellular respiration. That's where we get our energy from. Galactose is also going to be used in lactose. Fructose is corn syrup, right? High fructose corn syrup. Plants make fructose. The way that you're going to remember this, you know what monogamy is? Like a couple that's only made out of 2 people, right? So if you are monogamous, mono, then you can have a good girlfriend or be a good girlfriend. Glucose, galactose, fructose. All right? For disaccharides, you will die if you like my status. By the way, what is lactose? Where do you find it? Milk. Does everyone have lactase? The enzyme to break down lactose? You do as babies, but as an adult, some of you lose the ability to continue to break down lactose because you lack lactase. Ergo, the lactose irritates your intestinal lining because you can't break it down further, and then you diarrhea all over the place. You're welcome. I'm hilarious. Ha ha.
You have to be what's the word? Like if you're told by a doctor.
Lactose intolerant?
Yeah. What's that called?
Diagnosed.
Yes.
Oh, there's so many people who are not diagnosed. And it can develop later in life, which is something to look forward to as you age into your deaths. Okay, maltose. What is maltose? Where do you find it? Beer. It's also in malted beverages, and if you like Whoppers, the sugar in the Whopper is actually maltose instead of regular sugar. Sucrose. Sucrose is a fancy name for something you probably have in your kitchen. Just table sugar. When you use sugar, it's sucrose. Okay? So again, monosaccharides. If you're monogamous, you'll have or be a good girlfriend, unless you're gay and a male, in which case this isn't going to work for you. Like this acronym or mnemonic. Life will still work for you.
You could be a good gay friend.
If you're monogamous, you can be a good gay friend. I don't know what's happening in my world. Speaking of things I probably shouldn't say to students, you will die if you like my status. Oof. And then lastly, this is my favorite, just picture a pirate, right? Polly gets crackers. Sometimes.
What is the S of?
Sometimes. So Polly gets crackers sometimes. Poly, glycogen, cellulose, and starch. Okay. Questions, comments, concerns? I'm stopping. Maybe.